Hello, my name is Dr. Charlie Santerre. I'm at Purdue University. Today we're going to talk about foods enhanced through biotechnology. And I want to answer the question, are these products safe for consumers? So in this slide, you'll see that many of the foods that we eat today came from different parts of the world historically. So if you look here in South America, we've got peanuts and potatoes. Corn came from Central America. If you go all the way over to Asia, you see something that we grow a lot here in Indiana and other parts of the Midwest, soybeans. Is not even a native crop to the United States. It came from Asia. So over the last 10,000 years, we've crossed these crops and improved them and made them what we find today. Let me demonstrate that. In this slide, you can see what the original corn looked like. It was called teosinte. These are the little spikes of corn that you see in the middle of the slide. And a US penny is at the top. At the bottom, you can see what we grow today. So over 10,000 years, humans have used conventional breeding, crossing a male and a female, to go from these little spikes of teosinte to what we consume today. It's been a marvelous achievement, and it's not been overnight. It's taken quite a long time. So the challenge that we face is that the world population, as you know, has increased. So between 1960 and 1990, the population on the planet doubled from about 2.5 billion to about 5 billion. The challenge that we faced at that time was, how do we feed an extra 2.5 billion people? And this is where a term called the Green Revolution came about, where scientists were able to increase the productivity of our farms in order to meet the demand of the time. And if you look even further, this is going to some World Health Organization statistics, you can see that it wasn't only that we were able to increase the acreage for our crops, but we also increase the productivity of our crops. So on the left graph here, you see that maize, or known as corn here, increased dramatically from 1960 to 2010. We've also seen great increases in yield for rice, wheat, and soybeans, and productivity has also increased. So productivity, being the output minus the input, has also grown steadily. So these marvels of agriculture production have really taken us to a much greater place than we were 30, 40 years ago. One of the benefits of these advancements in agriculture has been the fact that we don't spend any more of our disposable income for food. In the United States, we spend somewhere around 10% of our disposable income on food, and that's been pretty constant, even with the declines in the economy. It's amazing that we only have to spend 10%. Many countries around the world will spend 50, 60, 80% of their disposable income on food. So let's talk briefly about plant biotechnology. In essence, what we're doing here is we're going to take a DNA or a gene out of one species, and we're going to move it into our soybeans, our corn. This will add only a single or a couple genes to the 30 to 40,000 genes that are found in our soybeans or our corn or our other crop. Let me get a little more technical with the next slide. So the way this works, at the top of the slide you can see we start with DNA, which is found in the cell's nucleus. This is the double-stranded helix that Watson and Crick described back in the 60s. And from the helix of DNA, we transcribe and make messenger RNA. Messenger RNA leaves the nucleus, goes into cytoplasm, where it hooks up with the endoplasmic reticulum, and this is where proteins are built. So let me summarize this maybe a little bit easier. So DNA is the blueprint for any proteins that our bodies or any living organisms need to survive. So the blueprint DNA is transcribed to the RNA, which is then translated to produce a protein. It's a rather technical process. The reason we discuss it is when we genetically enhance a crop, we're going to add some DNA or genes into the nucleus, and these are then going to transcribe and add a new protein into that crop. So the bottom line is, you add some DNA, and at the end of the day, you'll have some additional protein in the crop. So how do we do this? Well, there are a couple techniques. I want to talk about one of the ones that's not typically used today, but kind of demonstrates what's happening. So the scientists who came up with this were geniuses or ready for the nut house. And they essentially made a gun, much like a shotgun, where they coated these little golden tungsten pellets with the DNA or the genes that they want to move into the crops. And then they fired these literally with a bullet charge. So they fired these little projectiles down into the plant cells that they wanted to modify. And some of the cells were totally destroyed. But some of those cells picked up the DNA from the pellets into their own nucleus. And then the scientists were able to create these new crops. So this is actually the technique, the gene gun is what it was called, the actual technique that was used to modify the early forms of Bt corn. Now, we don't like to have a lot of bullets in our labs anymore, so they've found better techniques for moving DNA into our plant cells. So what are the crops that are in the marketplace today? Well, let me first start by what started a lot of this revolution. And so back in 1980, the US Supreme Court made a decision that companies could patent their DNA. A biotech company could develop a new strain of crop, and they could obtain a patent for this. And this was critical, because if companies couldn't obtain a patent for the DNA that they've modified, they couldn't have any way to recoup their investment. The number of crops that we see in the marketplace today that have been genetically enhanced is not that extensive. Tomatoes, soybeans, corn, potato, cotton, rice, plums, papaya, squash, sugar beet, cantaloupe, canola, flax, radicchio, and wheat. So the list is rather limited. This is all that you get. Now, I often ask my students, what in the marketplace is genetically enhanced? Well, everything in the marketplace is genetically enhanced, but not using this technique. So I often ask them, what about Thompson seedless grapes? And they go, yeah, that's got to be genetically enhanced. And no, that was done by conventional breeding, where you take a male plant and a female plant and cross them, and then select for the traits that you're looking for. So let's spend a few minutes to talk about how some of these crops have been enhanced and what we've done to them to make them better. Now, let me start with the Flavor Saver tomato. So the scientists at Calgene were trying to figure out a way that we could allow tomatoes in the field to ripen, fully ripen, get nice color, nice aroma, nice flavor, without softening. Because if we let tomatoes in the field that we're going to machine harvest get fully ripe, when you machine harvest them, they're just going to turn into a tomato paste mixed in with the dirt. So right now, we wait till they're just starting to turn ripe, and we harvest them, and then we try to ripen them back in the processing plant. So what they did is they said, how do we slow down the softening of tomatoes? And so they took out the DNA that codes, the term they use is codes for this enzyme, polygalacturonase. They took it out and they flipped it over and stuck it back into the tomato. Now, what does polygalacturonase do? So this enzyme is produced as the tomato ripens, and it causes the pectin in the tomato to break down. When the pectin, which is the glue that holds the tomato cells together, is broken down, it softens, as you see during ripening of the tomato. So by taking that DNA, that gene out, and sticking it back upside down, it now produces an enzyme that does not soften the pectin in the tomato. So it was pretty creative. Flavor Saver tomato was approved in 1994, and this picture here shows how it can affect the shelf life of a tomato. So on the left you have your Flavor Saver tomatoes, and on the right you have your conventional tomatoes. Just storing these tomatoes, you can see that as they soften, it causes a rapid decline in the quality of the tomato. So this is one technology that might extend the shelf life of our products. Let's talk a little bit about Roundup Ready soybeans. If you live in any of the states that grow Roundup Ready soybeans, you see these commercials on TV and you wonder what they are. So I usually start this discussion in class by asking students, what's the problem with weeds in our fields? And students usually get it right. They say that weeds will compete for the sunlight and cause the soybeans not to get as much sunlight. They compete for the rain, the water that the soybeans need. They compete for the nutrients. And one of the things students don't often get is when you harvest the soybeans, if those weeds produce seeds that are about the same size as a soybean, it can also contaminate your crop. So what the scientists at Monsanto did is they genetically enhanced soybeans to express or to produce more of an enzyme that's natural in the soybeans. This enzyme is what is inhibited by glyphosate, the herbicide that I spray on my crop to kill the weeds. So what happens when I spray glyphosate or Roundup, onto my weeds, it prevents the weeds from producing aromatic amino acids. If the plants can't produce aromatic amino acids, they can't produce the proteins that they need to live. So when Monsanto modified this DNA in the soybean, it produces more of this enzyme, so that, and therefore the soybeans will survive while the weeds will die. So Roundup Ready soybeans were introduced in 1996. In the last growing season, 94% of all soybeans grown in the United States were genetically modified. Another popular crop is Bt corn. What does the Bt stand for? Bt comes from a common soil bacteria called Bacillus thuringiensis. This bacteria produces a protein called Bt that we've added to corn to kill insect pests. So when the insect feeds on Bt corn, it gets this protein into its gut, and this protein forms spears that punch holes in the gut lining of the insect. It's really kind of cool how it works. And so the insect then dies from infection. But now when you and I, or most other mammals, eat this, we don't have the same gut conditions as these insect pests do. So we just digest this Bt as a regular protein. So for us, it's a nutrient. For the insect pests, it's something that can kill them. So Bt corn was also introduced like soybeans in 1996. In the last growing season, 88% of our corn grown in the United States was genetically modified. So there we've talked about a few. Let's talk about one that I think is also pretty exciting that's only come out in 2003. To talk about this one, I want to introduce the Western corn rootworm. This has been called the billion-dollar pest because of the damage it causes to agriculture. So in this slide, you can see in the top right, this is extensively damaged roots that were caused by the Western corn rootworm. And in the lower left, you see healthy roots. So by adding another Bt protein to this that is expressed in the roots, the scientists have been able to create a crop where this insect pest will eat it. They'll get this Bt protein. It'll cause damage to their gut lining. They'll die of infection, and there's minimal damage to the corn. And so it's called the billion-dollar pest because that's what it costs agriculture to have this kind of damage to the cornfield. So just amazing what we've been able to do since the mid-90s. So let's change tact here and talk a little bit about how these things are regulated. So who regulates them? Who makes sure they're safe for us to eat, safe for the planet, safe to grow? So let's start with the planet. So EPA regulates pesticides. So they make sure before this Bt corn is grown that it is safe for the planet. USDA makes sure that it's safe for agriculture. They don't want anything going out into the fields or into the barns that won't be safe for agriculture. So USDA has that role. Relative to safe to eat, FDA and USDA share that responsibility. So USDA regulates meat, poultry, and egg products. And since there are no genetically enhanced meat, poultry, or egg products, USDA is really in the backseat on this topic right now. So FDA regulates, the Food and Drug Administration regulates everything else. So FDA is the federal agency charged with regulating genetically enhanced crops. So how do they do this? If I was a biotech company and I brought this new genetically enhanced potato to FDA and said, I'd like to get this approved to be grown in the field, and I set it on the table and it started to crawl away, FDA would probably want to sit there and have a little longer discussion with me, because the standard they use is that my genetically enhanced crop has to be substantially equivalent to those that we find in the marketplace today. So there can be no unanticipated effects from my genetic enhancement. It should not be crawling across the table. They also look to see if there's any additional naturally occurring toxicants produced in my genetically enhanced potato. They want to make sure that it has the same nutrient content as the potatoes we eat every day. They also want to know that it has not been made allergenic, and we'll talk about food allergies in a little bit. So it typically takes somewhere between 4 to 8 years for a biotech crop to complete an FDA consultation. Actually, in recent years, it's gotten to about 1.5 to 2 years. So it's a little bit quicker. So let's try to answer the question, are these crops safe to eat? That's an important question that consumers ask all the time. You're genetically enhancing these crops. I'm eating them. I don't know much about them. Are they safe to consume? Well, let me tell you a little bit of a story that I think will show why some of these crops may be even safer than the ones we've been eating for decades. To do this, I have to talk about mycotoxins. So what are mycotoxins? Mycotoxins are chemical toxicants that are produced by molds as a result of injury, in this case due to corn, from insects, drought, or physical damage. And here on the right you can see just an ear of corn that's been damaged. And you can also see some mold. I don't know if this mold specifically produces a harmful mycotoxin, but it could. So anything that hurts that corn plant in the field, whether it be drought, insects, hail damage, anything, wind damage, weakens the resistance of the corn plant to prevent molds from growing on the corn. And some of these molds produce chemicals that are pretty toxic to us. The one on corn that we're often concerned about is called fumonisin. So let me tell you about a study that was conducted at Iowa State University. In the top left graph, you can see number of infected kernels per ear of corn from 1995, 96, 97, 98. The blue bar in this graph is conventional corn. The red is BT corn. And you can see in each of these years, the number of infected kernels, so the amount of mold that's growing in the conventional corn is greater than what we find in the BT corn. In the lower right figure, you can see fumonisin, the actual chemical that's produced by the mold that can grow in the corn, comparing conventional corn in blue bars to BT corn, in this case, in the yellow bars from 1997 and 1998. And so there's a significant reduction in this toxic chemical produced by molds in these 2 years. And here's what the Iowa State folks think is happening. That if you can prevent insect damage on your corn, you'll have less mold growing and you have less mycotoxins. Because BT corn produces a protein that these insect pests can eat, and it kills them before they can cause much damage. There's less damage to the corn, and then there's less mold that can grow. So in this exciting example, you can see that this technology can actually make our foods safer than what we've been consuming for hundreds of years. Here's another example I think you'll find pretty exciting. And it relates to the amount of pesticides that we put on our fields. To talk about this, I need to talk about BT cotton, another crop that was introduced in 1996. So look at this graph in the center here, and you can see from 1986 to 1995, the number of times cotton producers had to go into the field to spray insecticide to control for bollworm and budworm. And you can see they had to go in 5, 6 times to spray insecticide. When BT cotton was introduced in 1996, you can see the number of sprayings that had to occur in the field dropped to about 2 times per year. This means that pesticide reductions occur when we use BT cotton, BT corn, other BT crops. And it's a pretty exciting story. So what has been the pesticide reduction from 1996 to 2011? We've applied 8.7% less pesticides on our crops, which is about 197 million pounds of pesticide that we haven't thrown into our fields. It's a pretty exciting story. Now you might ask, why did I bring up cotton? We don't eat cotton. Well, actually we do. We eat a lot of cottonseed oil. So while we use cotton for our clothes, we also use cottonseed oil for our food. In 2011, 90% of cotton was genetically modified. So in that presentation, we talked about what a genetically enhanced crop is, how it's modified, how it's regulated, and also some of the advantages of food safety and environmental protection that we see from these genetically enhanced crops. It's a pretty exciting story. So that was the end of our Module 1. We have a second module that will continue this discussion and show you some of the exciting things that these crops can bring to us.